our learning objectives, which you saw in the course catalog or on the website when you chose to attend this course. We're going to really work through some differentiation between the two current versions of RECIST, 1.0 and 1.1. We'll talk about tumor data and the components of tumor data that feed into actually RECIST response evaluations. We're going to give you the opportunity to practice actually calculating disease response. We do have an activity for that, and we'll work through an activity together in the slide deck itself. And we do want you to also be able to identify and predict maybe some trends with tumor data, some challenges that you may encounter in working with RECIST data. And I'm hoping that you'll then be able to take some of those tips or common trends, and as I mentioned before, incorporate various aspects into maybe your case report forms or your protocol designs in order to mitigate that risk. Whenever RECIST criteria are used for response evaluation in our trials, it's really critical for all of the research team, project managers, medical monitors, CRAs, investigators, and site staff, to really clearly understand and consistently apply the criteria and the decision-making that is actually incorporated into RECIST. Obviously, a failure to do so could really put our study objectives in jeopardy. So that's the whole reason why we're here today together, to try to kind of wrap our brains around how you use RECIST and how you can ensure that the data coming out of your sites is reliable. So as I mentioned before, this is what RECIST stands for: Response Evaluation Criteria in Solid Tumors. So is there anyone on the line that's not working with solid tumors? You may be working with a hematologic malignancy or something of that nature. If your team is working with a different type of oncology trial, just give me a green check so that I can sort of customize the content a little bit to your needs. Doesn't look like it. Okay, excellent. So RECIST is a standard way to measure, essentially, in the simplest possible terms, how well a cancer patient is responding to the treatment within our clinical trials. It was developed initially through an international collaboration, and I want to read to you the National Cancer Institute definition, really, of RECIST criteria. It's a voluntary international standard, not just an NCI, a National Cancer Institute standard, so not just a U.S. standard. RECIST criteria are based on a simplification of some former methods that the WHO and then the ECOG consortium had come up with some other sorts of response criteria before RECIST was developed. And the reason RECIST is different is because it's really based on measurable disease. So NCI, makes it very clear that the benefits of RECIST are that you're actually looking at measurable disease. So we're trying to get to some objective, standardized measurements. So how has RECIST positively impacted the conduct of solid tumor trials? They really have been widely adopted throughout the world because of its foundation. RECIST gives you a foundation of some validated reproducibility. It really is a very reproducible type of criteria that you could have, all things being equal, you could have multiple radiologists looking at a CT scan or an MRI scan or what have you, and if they apply the criteria, they'll generally come up with pretty much the same sorts of evaluations. It's also been accepted broadly as a surrogate for survivability endpoints in clinical trials. And that means that regulatory authorities are very welcoming and accepting for you to use RECIST criteria and RECIST response evaluations as a predictor, if you will, a surrogate biomarker, if you will, for survivability. So you may not have to actually extend your protocols out to 5 or 7 year survivals. The regulatory authorities may instead accept complete response, for example, evaluations as obtained from RECIST. So the use of RECIST in our clinical trials really lends credibility to our endpoint data from the perspective of regulatory authorities academic institutions, industry sponsors, and actually the clinicians, the investigators who are seeing these patients. So I want to point out just the third bullet point here. It really provides us with an ability to assess in a quantitative manner whether a patient's disease has progressed or responded, whether a patient's disease has remained stable, and it allows us to look at both complete response and partial response. So we'll get into those details a little bit further in our time together.